Hey guys, Danny here. So in my last video, I kind of did a sweeping overview of what science is. So now I'm going to start getting a little more specific and in-depth. In this video, I'm going to delve more into the idea of the scientific process and the idea of science as a systematic concept. Prior to the 16th and 17th centuries, science as we know it didn't really exist. Yeah, it's true that many fields we know today existed in some sense. Medieval alchemists attempting to turn worthless raw materials into gold is kind of reminiscent of modern chemists, and the basis of mathematics goes all the way back to the ancient Greeks and Egyptians. However, it wasn't until these topics became structured that science emerged. There's a lot of debate on who specifically created what we now call the scientific method, and I'm really here to throw my hat into that debate, though it's most often argued that Sir Francis Bacon is the main father of the scientific method. All I can really tell you is that many early scientists contributed by developing systematic models and methods for analyzing the world at that time. Newton and Leibniz's developments of differential and integral calculus, the development and verification of the heliocentric or sun-centered model of our solar system, the development of Newton's natural laws, these all contributed to and were molded by the development of the standardized scientific process. Now to actually talk about this process a bit. First, a researcher is going to have to figure out specifically what they're trying to research, usually by asking a question. This generally begins with reading about what other scientists have published and done already, or using their own previous work to develop a question. It's an important aspect within science to be able to build off of someone else's work. Being able to develop these kinds of questions is one of, if not the first thing they look at and teach you when you enter grad school, and that may or may not be why I'm here instead of in a PhD program. Anyway, this standing on the shoulders of giants mentality is the first step, and generally you want to try and ask a question that hasn't already been answered. But you may also be interested in looking at one that has supposedly been answered, but in a different way. These don't have to be big, sweeping questions like where did the universe come from? You want something that you can answer through experimentation and observation. Though it's not unreasonable for a scientist to work on a longer-term project seeking to answer a broader question. Generally, for individual research, specific questions are better. As an example, which I'm going to continue to go back to in this video, let's look at the research that I did for my undergraduate thesis project. Rust tide, or Cotyledonium polycricoides, is a species of harmful algal bloom that occurs annually. It kills fish and shellfish by releasing a toxin that enters their gills and degrades the tissue. After reviewing the research that's already been done, I found while there's a decent amount of research concerning its life cycle and distribution, ecology, and most importantly, how easily it kills fish and shellfish. However, there was little research looking at how fish are affected when they are exposed to non-lethal or in what I call sublethal concentrations. There's also a decent amount of research looking at adult fish, but a lot less looking at larval. So after consulting with my advisor and considering what I as an undergrad could conceivably accomplish in the time I had the basis for my research. What effect do sublethal concentrations of rust tide have on larval fish? Next, a researcher needs to develop and consider a hypothesis. A hypothesis is essentially a proposed answer to your question. This is based on expectations developed based on previously published research. In my case, my general hypothesis was that larval fish exposed to these sublethal concentrations would have a reduced feeding ability and rates. This was based on the fact that in what little previous research on sublethal impacts existed, it was suggested that larval fish still experience impact. It's important to note, however, that a hypothesis cannot be proven, only rejected. A hypothesis can be supported by the evidence, and if enough strong evidence supports it, it can become a theory or a law. However, the very nature of a hypothesis prevents just, blindly accepting it. This also requires the creation of one or more null hypotheses. Generally, these are essentially the opposite of your hypothesis. So in my case, if my hypothesis is that there will be an effect based on sublethal concentrations, the null hypothesis would be that there won't be any significant impacts. So the goal then is to either reject your hypothesis or your null hypothesis based on the results you obtain. The real meat comes next, in the actual development of an experiment and the actual running of the experiment. There's thousands of methods that have been created for an infinite number of fields, and over time I hope to talk about many of the more commonly used and abused ones, but for now I'll just give an overview of what should always be practiced. When designing an experiment, you want it to have a few characteristics. First of all, it needs to be actually testable. No questioning how something happens in space if you don't have a space permit. In my case, all I needed were fish, water, shrimp for food, and the algae. Easy. You need to be able to exercise as much control as possible and limit the number of variables to what you're testing. In my case, all the fish were the same age, water parameters were the same, and basically everything was the same except for the things that I were manipulating, which was the concentration of algae in the water. This also includes having one or more controls, which account for the possibility that other factors may be at play. For example, I needed two controls, one that contained no algae at all, and one that contained only water and the food that was used to grow the algae in, called GSE. And this was to make sure that neither anything in the normal seawater or anything in the growth medium was having an impact. These factors can sometimes be a challenge depending on the kind of research being done, so this is a very important consideration when analyzing research that you're reading. You also want to make sure that you have enough replicates. Essentially, you want to repeat your experiment as many times as physically possible. The more data you have, the more trustworthy your results will be. This is another important factor to consider when reading research. Lastly, you want to make sure your research can be repeated by other people. In my last video, I mentioned that psychology as a field may be experiencing a replication crisis, which can significantly impact the validity of the conclusions that they make. In the end, I wound up with two main experiments for my research. In the first, I exposed fish to my 4 different concentrations for an hour and gave them 20 shrimp to eat, and determined the percentage they ate at the end. In the second, I gave them 10 shrimp and looked at how long it took each fish to eat all 10. After collecting all my data, it was time for analysis. Statistics is its own large field, and there's a lot that can be done in regards to your data in order to manipulate statistics. And unfortunately, it's harder to be aware of this kind of thing when reading research, so I hope that over time we'll be able to help with this, but for now all I can say is this. Statistics are tremendously important in interpreting results. No matter what method is used, it's very difficult, if not impossible, to draw any significant conclusions from data without statistics. As much as we'd like to just eyeball it, that's not the best thing to do. That being said, graphs are incredibly helpful. They allow for relationships between data to be visually seen and can be used to at least take a guess at statistical relationships and to interpret if what a scientist says seems on the level. Generally, statistical results will be stated in terms of statistical significance. This basically means that the difference between two data sets was significantly different enough to draw conclusions from. This is often represented by a p-value, which is the likelihood that the results obtained were just random chance. Generally, you want your p to be less than 0.05, meaning that it's 95% likely that your results mean something. In my case, this statistic said that in both my experiments, the highest concentration had a significant impact on the number of shrimp eaten by the fish and the time it took them to eat all 10. This leads to the interpretation of the results. This includes both the immediate interpretation of what the data means. In my case, that sublethal levels likely impact feeding behavior, but also the broader picture. In my research, for example, this is a larger ecological perspective. Rust tide blooms are highly concentrated in the middle, but the outer edges may have concentrations similar to what I used. Previous research has shown that larval fish exposed to these concentrations have trouble swimming after exposure. These means that these fish become essentially sitting ducks in these blooms and will be inhibited in their ability to eat. The fish used in my experiments were forage fish, which means that they serve as important food for other more important species and more charismatic species. It's become important nowadays to consider this bigger picture, not just because of the importance to the rest of the given field, but also in order to justify the research to those funding it or supporting it, or just people in the general public. The next step is basically to repeat the process. The goal by the end is that new questions have been generated by this research. Perhaps your question was answered but created new questions, or maybe your question wasn't answered and you may have a new hypothesis. Or in my case, your question was answered but you want to expand the scope. Science is a cycle of questions and answers to generate knowledge. Ancillary to this last step is the publication of this new knowledge into the larger scientific community. Generally, scientists will publish their research into a scientific journal, and the more astute of you may have noticed the similarities between a journal article and the scientific method. However, other options exist. For example, while I haven't published my research and probably won't on my own, I presented it at a few conferences, a notably terrifying experience, but a necessary one. Other scientists may publish books or other works. The most important thing is to make this knowledge viewable to everyone, or depending on where you go, everyone who can pay. Anyway, that's it, guys. Thanks for watching. As always, we accept any discussion in the comments or any corrections on mistakes we may have made. But see you next time.